Hello, biological anthropology lab students, and welcome to this tutorial on Punnett squares. In this video, we will learn how to do a Punnett square, and we will also learn how to calculate the genotypic and the phenotypic ratios that result from the Punnett square. So first, let's define what a Punnett square is. This is a method of determining the potential genotypes and phenotypes of offspring resulting from two parents. Just as a refresher, the genotype is the genetic coding for the trait. We are also going to be learning how to calculate a genotypic frequency or ratio, and this is the numerical representation of the proportion of homozygous dominant individuals, heterozygous individuals, and homozygous recessive individuals. Another definition that we should know already, the phenotype is the outward physical expression of this genetic trait. We're also learning our phenotypic frequency or ratio here. The phenotypic frequency or the phenotypic ratio is the numerical representation of the proportion of those with the dominant and with the recessive form of the trait. So let's put this into practice. How do we do a Punnett square? First, let's cross two parents, and the trait that we are going to perform this Punnett square with is going to be tall versus short. So we are looking into the height of the pea plant. So we have two heterozygous parents here. Heterozygous, again, means that we have one dominant allele, one recessive allele, forming this total genotype of big T, little t, otherwise known as heterozygous. So one parent here is heterozygous. Our second parent here is also heterozygous. So first, let's cross these two big T's. So these are, again, the dominant allele. So one big T is going here. This big T is also going here. And that is going to give us a genotype of homozygous dominant, or big T, big T. Next, let's cross this little t, so the recessive allele, with a dominant allele. So this combination is going to produce a heterozygous individual. So again, we have a big T and a little t there. This next cross is going to produce another big T, little t, another heterozygous individual based on this pair here. And last, these two little t's are going to give us a homozygous recessive individual. So we have, again, two little t's there. So we have completed one Punnett square, but let's figure out what our genotypic and our phenotypic ratios are. So here we have our Punnett square once again. The genotypic ratio, as I mentioned, is going to measure how many homozygous dominant offspring there are, how many heterozygous offspring there are, and how many homozygous recessive offspring there are. So based on this Punnett square, we can see that there is one homozygous dominant, there are two heterozygous, and there is one homozygous recessive. So that genotypic ratio is displayed here. On our quiz, so on our first quiz in this class, you will need to know how to calculate the genotypic and the phenotypic ratios. There will be two questions in particular on the quiz that ask you to do this. So you're going to have to do a Punnett square like this, and you will also have to calculate the genotypic and the phenotypic frequency. So let's get to that phenotypic ratio now. You see that I have 3 to 1, meaning that I have 3 individuals having the dominant form of that trait. So 3 individuals are going to be tall, 1 individual will be short. So over here you can see 3 tall individuals, 1 short individual. These are going to be calculated with ratios. So when you're doing this on your quiz, make sure that you write this in ratio form. With the genotypic ratio, make sure that you are doing homozygous dominant first, heterozygous, and homozygous recessive last. With the phenotypic ratio, make sure that we are starting with the dominant trait. Next, we will follow with the recessive trait. Let's try this one more time. So now we have, again, two different parents. One of them is tall, one of them is short. So the tall parent is homozygous dominant, meaning that they have 2 copies of the dominant allele. The next parent is homozygous recessive, meaning that they have 2 copies of the recessive allele. So let's start crossing these two parents. First, let's cross this big T with this little t, that's going to produce a heterozygous individual. The next pair, you'll notice somewhat of a pattern because we have another heterozygous individual, a third heterozygous individual, and a last heterozygous individual. So in this Punnett square, all of our offspring are likely going to look the same. So in this trait, they are all going to be tall and they're all going to be heterozygous. So let's get back to that genotypic and the phenotypic frequency. So the genotypic frequency, you're going to notice that there are no homozygous dominant individuals. Make sure to write that down. Make sure to write in your ratio that there are 0 homozygous dominant. There are 4 heterozygous individuals, so all 4 of these here are heterozygous. And last, there are 0 individuals that are homozygous recessive as well. So none of that here. Make sure that we indicate that also in our ratio. So if I were to ask you to do this pair, in particular on our quiz, this is what the genotypic frequency or ratio would look like. Now let's get to our phenotype. So you'll notice that all of them are tall because they are heterozygous. So we have 4 tall individuals and 0 short individuals. Again, this is how this should look on your quiz when you are doing this Punnett square and calculating these frequencies. I hope that this was helpful. Thank you for watching this video, everybody.